Hello everyone, today we'll discuss about transcription. So let's see what is transcription. Transcription is nothing but a synthesis of RNA molecule from the DNA molecule. Let's see how it is synthesized. Okay, so here you can see there are two strands of DNA. One we call 5 to 3 prime and then another one is 3 to 5 prime. The 5 to 3 prime strand is known as coding strand or also known as sense strand, while the opposite strand known as template strand or antisense strand. When the RNA is synthesized, the RNA molecule is complementary to this template strand. That is why we know it as a template strand. And the RNA molecules look like these coding strands. That is why we called that is why we known as a coding strand. Okay, so first let's understand the structure of gene. So in the structure of genes, there are three main elements are involved. The first one is a promoter region, then the coding sequences and the terminator regions. In the coding sequences, the ORF sequences is found. ORF is nothing but it's an open reading frame which codes for the amino acids or which synthesize the proteins. Okay, so what happened in transcription? So in transcription there are three major steps. The first one is the initiation, then elongation and termination. What happened in initiation? In initiation, the RNA polymerase or the transcription factor recognize the promoter sites on the DNA molecules wherever the gene is present, and after that the RNA polymerase comes and this elongate the chain and or synthesize the RNA molecules by using the template strand. And then here you can see after the synthesis of this RNA molecules, it get terminated and this the transcription get stopped. Okay, so in case of transcription, there are some enzymes involved that is known as the RNA polymerases, and there are different RNA polymerases in prokaryotes and in eukaryotes. So in case of prokaryotes, there are multiple subunits are combined and together form it as a holoenzyme, and this holoenzyme work as a RNA polymerase. Okay, so what happened in this holoenzyme? There are multiple subunits are involved. Like you can see here, there are 2 alpha, 1 beta, 1 beta dash, 1 omega, and sigma factor. Okay, so except this sigma factor, this other unit are known as together this unit known as a core enzyme. And when we combine the sigma factor with this, we known as a holoenzyme. Okay, so let's see the function of these subunits. Okay, so the function of these subunits in case of prokaryotes, the important function is of we can say the sigma. We also known as a sigma factor. The sigma factor recognizes the promoter sites. Wherever gene is present, the sigma factor will go and recognize that the promoter sequences or signals, and that there it binds and recruits the other RNA polymerase subunits, and then the transcription takes place. So there are different subunits and having different function. So like you can see here, both alpha unit that determine the DNA which is transcribed, and then beta one is catalyze the polymerization. It means the beta subunit is a main subunit which elongate the chain. Okay, then other subunit like beta dash, it's unwinding the DNA, and the omega, there is no function, there is not known function for omega. Okay, so you have to memorize this. This is the important one, the sigma factor, and this is the beta one, because the sigma factor recognizes the promoter region, whereas the beta catalyzes the polymerization. So these are two important things. Okay, so let's first understand what are the promoter sequences. Okay, so in promoter sequences, there are different promoter sequences in prokaryotes and eukaryotes. Okay, so in prokaryotes there are two major sequences. One is called Pribno box and another is called at -35 region. So this is located at upstream region. You can see there are two strands, coding strand and template strand. So what happened? This is the transcription start sites or TSS site. Mean this is +1, and upstream to this like -1, -2. Here you can say -10. So at -10 region, there are one like Tata box is present that is called as Pribno box. Okay, Tata sequence is present that is called as Pribno box, and we go further. Then at -35 region, there is another sequence is present that is also known as that is itself known as -35 region. Okay, so these two are the major elements of promoter in case of prokaryotes. Here you can see the how this promoter sequence is called in the box, and the little description are here. You can go through it. Okay, so let's start. Let's know about the termination process in prokaryotes. The initiation and the elongation is very simple, and we have to understand the prokaryote termination in prokaryotes. So to understand this, there are row protein are involved. We are also known as a row factor. Okay, so there are two processes, two different mechanisms are present in prokaryotes. The one is called row independent and another is called row dependent. So what happened in this, like see. So in case of row independent, when this RNA molecule comes to end, okay, at that time, so there are multiple inverted repeats. So these repeats form a these inverted repeats form a secondary structure with the help of the hydrogen bond. Okay, so with the secondary structure in the form, there is no secondary structure, there is no means repeats, or there is no complementary sequence. So there is here it is forming loop, and it's look like a hairpin. So we call it as a hairpin loop. Okay, so this hairpin loop triggers helps to dissociate the RNA polymerases, and then transcription get stopped. Okay, so this is we called as a row independent. Why we call it row independent? Because there are no row protein involved, so we know it as a row independent. That's very simple. Okay, so let's see the another one, that is row dependent. Row dependent means there is a row protein involved in the termination process. So in that case, so there is you can see there is a row protein, and this transcription, this is nascent RNA. Okay, so when the transcription is about to complete, this nascent RNA pull by this row factor, or it also help in the to dissociate the RNA polymerases, so that the transcription can stop. Okay, so this is very simple. Here it is a row protein, and it is pulling this nascent RNA molecules, and in this case it is forming a hairpin loop, so that the transcription cannot go on. Okay, so this is all about the prokaryotes. So there are some differences between the prokaryotes and eukaryotes promoter. Okay, so let's see the eukaryotic promoter. This is also important. So here you can see the promoter sequences, and this is some downstream sequences. So here this is the 5 to 3 prime. Okay, so you just understand one by one. Let's understand this one. Here is a +1 site. This is also called a TSS site, means start of transcription. And this TSS site, this is +1 site, and upstream to this there are Tata boxes present at -25. Likewise there is the Prigno box is present. So here it is a Tata box is present at -25, and this Tata box and this +1 region form a core promoter. So let's understand this with the help of this diagram. So it is highlighted here. So in the core or basal promoter, we can say there are three elements are involved. The one is the Tata box, known as a Tata box because there is sequence TA. Okay, and then another sequence is called as a BRE sequences, that is recognized by this transcription factor, and the TSS, that is transcription start site, that is +1. So these all three elements form we can say they are the core promoter elements, or we can say basal promoter. Okay, so this Tata box is recognized by this TF2D. Okay, so this is very important, we have to memorize this. This TF2D, it is associated with the Tata binding protein, so this bind with this Tata box, and first in the first process of the firstly this TF2D recognize this Tata box, then this TF2D recruit the other transcription factors like TF2B and others. Okay, so you have to memorize this. The Tata box is recognized by this 2F2D transcription factor. So you have to memorize this one, TF2D and TF2B. Okay, and then +1. These all together form a core promoter. Okay, so let's go to downstream. In downstream there is a +1 regions, means here it is start. So in +1 region, you know that in eukaryotes there are some introns are present, and prokaryotes there are no introns. Okay, so you can see this is the coding region, like exons are coding region and introns are non-coding region. Okay, so in between these two, when it starts, there are 5' UTRs called as a 5' untranslated region. Okay, and in 3' it is called as a 3' untranslated region, or 3' UTR. Okay, there are some function associated with this. Okay, we'll understand it later. And let's go next slide. Okay, so there are some enhancer sequence also involved. I haven't discussed about this. So these are some regulatory elements, proximal control elements. So these can enhance the transcription process, then these can upregulate or downregulate the transcription process. Okay, the mRNA. So here some are GC box or cat box are present. Okay, so apart from this, there are some enhancer sequence. These enhancer sequences, like this, you can see here, there is a gene, and this enhancer sequence is located apart from these genes. Here, this can be a kilobase pair away from the genes. Okay, so from there they can influence the transcription process. They can form a loop like this, and they can come contact to the promoter region, and they can able to influence the transcription process, or they can increase the rate of transcription. That is why we known as enhancer, because it is increasing the rate of transcription. So they can, even they are located far from this gene, they can come contact to this promoter region, and they can increase the rate of transcription. That is why we know it as enhancer sequences. Okay, so as I explained, in case of prokaryotes, there are some RNA polymerases. Likewise, there are in eukaryotes, some RNA polymerases are involved. Okay, so mainly there are three type of RNA polymerases are there in eukaryotes. The RNA polymerases 1, 2 and 3. Okay, let's understand first this. Okay, so RNA polymerase 1, 2 or 3. Okay, so these all are located in the nucleus, but except this RNA polymerase 1, because it is found in nucleolus. Okay, so you remember this, please. And here you can see the RNA polymerase 1 transcribe the rRNA. Okay, ribosomal RNA, transcribe the ribosomal RNA except 5S RNA. So except 5S RNA, other ribosomal RNA are transcribed by the RNA polymerase 1. Okay, while the 5S RNA are transcribed by the RNA polymerase 3. You can see here. Okay, so let's go one by one. So RNA polymerase 2 here, and this transcribed the pre-mRNA, which is messenger RNA, or also transcribed the siRNA, small nuclear RNA, siRNA, small interfering RNA, on the another is one the miRNA. Okay, that is microRNA. So miRNA, siRNA and messenger RNA are transcribed by this RNA polymerase 2, and RNA polymerase 3 is transcribed this tRNA molecule and 5S RNA. You just see here, this RNA polymerase 1 transcribed all ribosomal RNA except 5S RNA. So this 5S RNA are transcribed by this RNA polymerase 3, and some other nuclear RNA are also transcribed by this RNA polymerase 3. Okay, so these three RNA polymerases are present in the case of eukaryotes, but some other RNA polymerases have been also found. Okay, so these are the RNA polymerase 4 and 5. So these are present in plants. Okay, so 4 is this 4 is transcribed to this siRNA, that is called small interfering RNA. This RNA polymerase 5, this also codes for siRNA as well as some antisense RNA. Okay, so this chart is very important. Okay, so let's finish this. Thank you very much.